Hello everyone, I am Ms.S.Madhura, Assistant Professor, Department of Biochemistry, K.S.Rangasamy College of Arts and Science, Tiruchengode. Today we are going to discuss about cell membrane and its various model in this video. Now we are going to see the introduction of this topic. Before entering into our topic, we are going to see about what is cell. Cell is the fundamental unit of life. Cell was discovered by Robert Hooke in 1665 from the slice of a cork. Cell is divided into unicellular and multicellular organism. Unicellular cells are called as prokaryotes and multicellular cells are called as eukaryotes. Cell has different cell organelles and has various functions of the organelles. In the organelles, cell membrane is the vital organ which has a significant function in it. Each and every cell, prokaryotic and eukaryotic cells, have a thin layer of lipid membrane which is surrounded the cell is called as plasma membrane or cell membrane or cell lemma. These cells have the major functions like protecting the inner and outer layers of the cell, which allows the transport of molecules from inside and outside of the cell, and also the cell membrane maintains and checks the chemical substance produced by the cellular metabolism inside the cell, and it also exits the toxic substance which are produced by the cellular metabolism as a byproduct. Cell membrane has the thickness of about 70 to 100 area of thickness, and it contains proteins, lipids and glycoproteins. Now we are moving to history of cell membrane. At first early stages, Carl W. Nageli 1817 to 1891 described that there is a membrane surrounded by the cell which is made up of a membrane called plasma membrane due to the osmotic and other phenomenal functions of the cell. At first, before 1885, he named it as a xelenal membrane because of its nature. After 1885, he named it as cell membrane due to the discovery of an injured cell which possesses the capacity that it does not mix the cytoplasmic content into the watery content of a cell. This chart shows the history of the findings of the cell membrane. At first, Overton described that the cell membrane is a lipid layer. It is a single layer, and after that Lamangure described that the cell membrane is made up of lipids, and after that Gotter and Grendel said that lipid membrane is a bilayer, and after that Davidson and Daniel described about the protein structure present over the cell membrane, and after that Robertson described the model of unit membrane of a cell membrane, and the final conclusion model of fluid mosaic model was described by Singer and Nicholson as a fluid mosaic model. So we are going to see about the models of cell membrane. We have lipid bilayer model, protein hypothesis. In that we are going to see about two models. One is sandwich model and another one is unit membrane model, and then we are going to discuss about fluid mosaic model. Now we are going to discuss about lipid bilayer model. At first, this model was explained the structure of the plasma membrane which the cell was surrounded by a cell membrane. So previously no direct information about the cell membrane was on the history. After this discovery only we are discussing about the cell membrane which has a bilayer and it has a lipid content. So 1902 Overton observed that substance soluble in lipid passes easily from one side of the cell membrane to the other side of the cell membrane. So he concluded that the cell membrane is made up of a lipid. So after that Gotter and Grendel in 1926 observed that extract of the erythrocyte cell membrane has twice the content of a cell. So they discovered that the cell membrane is made up of a bilayer. It's not a single layer. So this does not properly exhibits the model of the cell membrane, but it laid down the foundation for the further model of the cell membrane. So now we are going to discuss about protein hypothesis. This hypothesis describes about the cell membrane which has a protein which are embedded or packed around the cell membrane. So it has a protein which act as a mediators. It functions as a receptors or protein molecule which facilitate the transport of substances from inside to the outside of the cell membrane or outside to the inside of the cell membrane. So the layer is made up of a lipid, that is a bilayer, and it also discusses about the asymmetric arrangement of the lipids and proteins over the membrane. It also conveys that there is an interdependence between the protein and lipid substances which are responsible for the fluidity of the membrane. So we are going to discuss about Daniel and Davidson model. So this model is also known as sandwich model. This model describes that the cell membrane has a lipid bilayer which has a lipid head and tail portion. The head portion is hydrophilic in nature and the tail portion is hydrophobic in nature. Hydrophilic means it accepts the water molecules. Hydrophobic means it repels the water molecules. molecules. So then when coming to the protein coating area, this model explains that as a protein is covered over the cell membrane, which acts like a sandwich in the model. So after membrane structure, they have said that the protein are not embedded over the membrane, it is only packed over the cell membrane. So then limited protein role. This model also does not describes many vital roles of the protein in the lipid bilayer. So this is the picture of the sandwich model proposed by Daniel and Davidson. This has the hydrophilic zone of proteins which interact with the hydrophilic head portion of the lipid bilayer. So next we are going to see about unit membrane model, which was proposed by Robertson in 1959. This is similar to the sandwich model, but this has some unique points over the model. Unit membrane model is the fundamental model for the fluid mosaic model. So there is only some corrections in the fluid mosaic model which is accepted worldwide. So unit membrane model has described that lipid bilayer has the inner hydrophobic nature lipid content and outer head portion of a hydrophilic nature. Then the protein layers are present over the membrane, and then it describes about that from which they have concluded that it is a unit membrane model by using the electron microscopy technique, which replicates the 3 layers of the cell membrane, which has 2 dark electron dense layer and a single electron lucent layer, which describes that dark layer is considered as the protein substances and the light layer is considered as the lipid substances. So this concluded that every living organism in the world has cell membrane and its functions only differ. Mostly it has the universal framework of the structural membrane. So and also it is considered as the simplicity structure of a cell membrane, which in detail furtherly discussed in the fluid mosaic model. So now we are going to see about fluid mosaic model. Fluid mosaic model was proposed by S.J. Singer and A.L. Nicholson in 1972. According to this model, lipid bilayer is made up of 2 lipid content which has a head portion and a tail portion, and it describes that the proteins are not sandwich like packed membrane. It is embedded in the cells and it is projected out from the lipid layer. So from this model we have concluded that membrane proteins are not fixed or not overly coated over the cell membrane. So phospholipids and the glycoproteins have the nature of hydrophobic, which is the responsibility of the fluidity of the membrane, and the globular proteins of the membrane are of 2 types. One is integral proteins and another one is peripheral proteins. The peripheral proteins are extrinsic in nature and integral proteins are intrinsic in nature. Then the rapid movement of proteins and lipid molecules are described in this model. Thus we have to say that lipid cell membrane is a flexible membrane which rearranges the lipids and proteins present in it. So this flexibility proposed by Singer and Nicholson describes that Davidson and Daniel has made a mistake in their proposal, that the lipid bilayer is not rigid or static. It is movable and the proteins and lipids are interconversible in the model. So proteins and membranes are considered that proteins in the lipid membrane has the enzymatic function which helps in the transport of substance by a particular channel, which allows the substance to move from one end to the other end. So the fluidity of the lipid membrane was discovered from the study of X-ray spectroscopy and after the electron spin resonance test. So we are going to now discuss about the dynamic properties of a lipid membrane. The dynamic properties are the lipid membrane is flexible and the content in the lipid membrane can move easily from one place to the another within its arrangements, and the lipid content can move in its axis also and rotate on its axis. So thus the fluid mosaic model was accepted worldwide as a correct model. Today we have seen about the cell membrane and its various models proposed by different scientists. I hope you all get clear understanding about the topic. Thank you.